Welcome to the problem-solving set on the operon concept. The first question is a fill-in-the-blank that says the structural genes of operons are of two types, those that are always transcribed and translated, termed blank genes, and those that are not but can be turned on, termed blank genes. So let's go ahead and fill in the blanks. For the ones that are transcribed and translated always, these are termed in genetics and biology constitutive genes. For the ones that aren't always transcribed but can be turned on, those are termed inducible genes. So we'll see in the next section on the lac operon, that is an inducible operon that has inducible structural genes, that they're not always transcribed but they can be turned on. Problem 2. Using the word bank below, match each statement with the correct term relating to the operon concept. A large mRNA molecule that includes the transcripts of many genes, also containing multiple stop and start codons. So this is what's known as a polycistronic mRNA. Now this is a piece of mRNA that, as it says in the statement, contains the transcripts of many genes. So it's not to say that the whole thing will be transcribed into one large protein. In fact, they have many stop and start codons within this mRNA so that you make multiple little proteins. And that's what, in the operon, the structural genes are usually grouped together so that when transcription occurs, let's say there's 3 structural genes in the operon, they'll all transcribe together and then they're translated into 3 separate proteins or enzymes. B. Genes found a distance away from operons code for regulators or repressors. So these are what's known as regulatory genes. So, for instance, as you'll learn in the next chapter, the lac operon, there's a gene for a repressor molecule that is away from the operon, so it's not in the regulation of the operon so that it can regulate it. It has to be outside of it. C. Protein that blocks transcription of an operon by binding to the operator. This is what's known as a repressor molecule. So these are proteins that, as it says, binds to the operator and kind of just is a roadblock so that RNA polymerase can't get past it and therefore can't transcribe the genes. D. DNA that encodes a polypeptide. Well, this, any DNA that's a gene can encode for a polypeptide. But for this, because we talked about regulatory genes in Part B, here this is talking about a structural gene. So usually in operons, the genes that are grouped together, the structural genes, all have to do with the same pathway. So for the lac operon, they all have to do with degrading lactose, and for the trp operon, they all have to do with synthesizing tryptophan. E. Enzyme whose synthesis can be inhibited by a metabolite that acts by activating a repressor. This is a repressible enzyme. Okay, and the reason for that is that it can be inhibited, and that's how you know it's repressible. F. A region of DNA that acts as an on-off switch for the operon by binding the regulatory protein, located between the promoter and the structural genes. So this is what's known as the operator. So we'll see in later questions the general structure of an operon. There's the promoter, the operator, the structural genes, etc. And the operator is actually the molecular switch is what a lot of people call it, because it can bind the regulatory protein, which is where you have that roadblock to RNA polymerase. G. Enzymes whose synthesis can be stimulated by metabolites that act by inactivating the repressor. These are inducible enzymes. And here the hint for this is that it's stimulated by metabolites, whereas in Part E, the keyword was inhibited. And finally, H, a region of DNA that binds DNA-dependent RNA polymerase. Now this is the promoter. It's another part of the operon. It's in the section that's known as the regulatory region, so it's near the operator. And the promoter, this is where DNA-dependent RNA polymerase binds and therefore can go down and complete transcription. Problem 3. Using the term bank, label the illustration of a simple operon. So this illustration is actually based on the lac operon, although you don't need to know the specifics for this question. So let's see. If you look at the far left, that red box that looks like it's outside of these other 5 boxes kind of grouped together, this one right here, this is outside of the operon. Remember from the last problem, the gene that is outside of the operon is the regulatory gene. So this usually encodes for a repressor, therefore, and it's outside of the operon so that its transcription isn't connected to the operon, so therefore it can regulate the operon. So usually it has its own promoter. Okay, the next region with these beige, the 2 beige boxes, combined, that is what's known as the regulatory region. Regulatory region. And this includes the promoter and also the operator. So remember, the operator is the on-off switch, and the promoter is where DNA-dependent RNA polymerase binds. So the first in this is the promoter is the first one, and then comes the operator. So when you have a repressor protein bound to the operator, let's say, I'll just draw it in black. Let's say you have a big repressor protein here, and it's bound to the operator. It's active, and it's trying to block transcription. Then I'll draw it in green. If RNA polymerase tries to bind here, I'll show it like that. If it tries to bind to the promoter, it can't get past that repressor. So this is the regulatory region, because depending on different metabolite levels, depending on which operon you're looking at, if the operator is bound with the repressor, RNA polymerase cannot transcribe the operon. Okay, so that's the promoter and the operator and the regulatory region. So obviously these last 3, the grouping, are the structural genes. And you'll learn the specific of the 3 structural genes for the lac operon in the next chapter. But generally, structural genes encode for the proteins that are involved in the pathway. So if it's a biosynthetic pathway, if it's a breakdown pathway like in the lac operon, then those are the genes that encode for those specific enzymes and proteins that the pathway requires. Problem 4. Damage to bacterial DNA can lead to the expression of many genes involved in repair mechanisms. A loss-of-function mutation in the PEP-X gene leads to the expression of these repair genes even when there is no damage to the genome. Would you propose that the PEP-X protein is a positive or negative regulator? Explain. So let's take stock of what they're telling us. They say a loss-of-function mutation in the PEP-X gene. So a loss-of-function, we could just obviously that means that the PEP-X protein isn't going to function as it normally does. So basically that means that we don't have PEP-X protein anymore. So without the PEP-X protein, it says that these repair genes, the DNA repair genes, are expressed even when there's no damage to the genome. So we're going to say that without PEP-X, the repair genes are constitutively expressed. Remember, that means that they are always expressed no matter if there's a trigger or not. Okay, so how would we explain that? So if the PEP-X protein was a negative regulator, this would explain that without it, the repair genes are constitutively expressed. So I'm going to write the answer is that PEP-X was a negative regulator. And then I'll put it in context so that you can understand. So let's say PEP-X protein equaled a repressor protein when it has a normal function. So remember that a repressor protein, when it's active, can bind to the operator of an operon and block transcription, because remember, RNA polymerase can't get past it to transcribe the structural genes that are downstream on the DNA. So if it was a repressor, that means that it would block transcription unless there was an environmental trigger like bacterial DNA damage. So it would block transcription until trigger of DNA damage. Now when you use this example as an explanation, if you use the explanation that PEPX is a repressor protein, that explains the result that without this protein, they're always expressed. Because think about it, if you don't have a repressor protein anymore, then RNA polymerase can just continue to transcribe this operon because there's nothing blocking it, even if there's no DNA damage to be had and there's no reason for these to be expressed. So you'll find this more and more, that prokaryotic regulation is really so that the cell doesn't waste any energy. So if there's no DNA damage, then there's no reason to express these repair mechanism genes, right? So that's what, in this scenario, this PEPX normal functioning gene helps, is that it represses these genes unless there is a need for them. Okay, so this is a negative regulator, and you can explain it by proposing that the PEPX was a repressor protein. Problem 5. Contrast regulation by repression and induction. So the first is repression, and you're going to learn this in 2 chapters, but this is an example of a TRP operon, the tryptophan operon. So I'll just write that here, the TRP operon. So the regulation by repression is when a metabolite, so in the TRP operon, that metabolite is the amino acid tryptophan. When a metabolite is added, it binds to the repressor, and when it binds to the repressor, it causes a 3D conformational change that actually, in this case of the repression, activates the repressor. So I'm going to say binds to repressor and activates. It's a key here. And when this repressor is now activated, it can bind to the operator and turn off transcription. So transcription turns off. Okay, so you add the metabolite, cell sees that it has the metabolite which binds to the repressor, and the repressor becomes activated, so therefore it can bind to the operator, turning transcription of the operator off, of the operon off. Okay, so this regulatory mechanism in prokaryotes is used for the purpose of, if you already have access to something, then there's no reason to waste energy to make more of it. So when a cell already has enough tryptophan, there's no reason to waste the energy transcribing the enzymes that are used to make tryptophan, right? Okay. Now for induction, on the other hand, a prime example that you'll learn in next chapter is the lac operon. So induction is where you add a metabolite, which is known as an inducer. So I'll put that in parentheses, inducer. And the inducer combines with the repressor again, so binds to repressor. But here's where it becomes contrasting. When this activator, the inducer, binds to the repressor, it actually inactivates the repressor. So before the metabolite was added, the repressor was active, was bound to the operator, blocking transcription. But now you've added in an inducer, which changed the conformational the 3D conformation of the repressor, inactivating it, and transcription now is turned on. Because when a repressor protein is inactivated, it can no longer bind the operator, and RNA polymerase has no roadblock on the operon. Okay, so this is under the idea that a cell doesn't want to waste energy making genes for utilizing something unless it's there. So the genes in the lac operon are used to break down lactose as an energy source, but the cells would prefer to use glucose. So if glucose is around, there's no reason to make these lactose utilization genes because they don't want to use it. So they'll only use these genes, and this operon is only turned on when lactose is the only option for them. Okay, so these are the difference between repression and induction, and then the next 2 chapters we'll go deeper into the details of the lac operon and the TRP operon, and you'll really see both the similarities between the operon concept in the whole and the differences between repression and induction.